Thanks for a great talk. Our next speaker is Mark Hahn from Woods Hole Oceanographic Institution, where he's going to be talking about applications of genome editing to understanding chemical sensitivity.
Thank you, David. Good afternoon, everyone. I'd like to begin by acknowledging my co-authors on this presentation, Neil Aluru, who's here at the workshop, and Sibel Karchner. What I'd like to do today is talk a little bit about some applications of genome editing for understanding mechanisms of developmental toxicity with a focus on factors that determine sensitivity or resistance to developmental toxicity. And I'm going to present a couple of examples of ongoing projects in the lab, which I'll describe briefly. And then at the end, I would like to talk about some challenges and some opportunities for genome editing and applying it to developmental toxicology. Now, thank you, Rebecca, for introducing adverse outcome pathways. As all of you know, one of the main goals in toxicology is to understand the connection between exposure to toxicants and adverse outcome pathways. Now, acting in concert with these adverse outcome pathways are factors that serve to modify the sensitivity of the pathway to the toxicant. And these include some well-known things like constitutive biotransformation activities, variations in the sensitivity of the target site, the molecular initiating event, inducible protective mechanisms, including drug metabolizing enzymes, and a variety of repair processes that exist in the cell. And we've heard about some of those today already. We're especially interested in understanding the function of proteins that can serve both as targets and as regulators of these inducible protective responses. One such protein, which Dave Scher introduced already, is the Aryl Hydrocarbon Receptor, or AHR. And in case you've forgotten, since Dave's talk, what the AHR does, it is a ligand-activated transcription factor. It binds with high affinity to dioxin-like compounds, including many PCBs and PAHs. Once activated, the AHR moves into the nucleus, where it activates transcription of literally hundreds of target genes. Many of these are involved in development or other physiological roles. Some of them are involved in adaptive functions like regulating drug metabolizing enzymes. And a number of others provide direct links to toxic effects, and these are not as well understood as the other two. So we're interested in understanding what factors influence the sensitivity of this AHR signaling pathway to toxicants. We do this using fish models primarily. As you know, fish are vertebrates that exhibit most of the same genes and pathways that humans have. They have a number of technical advantages, and all of this has led to fish being established models in developmental biology and increasingly in developmental toxicology. Now, one of the interesting features of fish is that they often have extra copies or duplicates of genes that exist as a single copy in mammals. And this includes the AHR, which exists as multiple copies in fish compared to the single copy in humans. Another interesting feature of fish is that many fish are exposed in the environment to high concentrations of chemicals, so they can serve as good models for population-level impacts of contaminants in a real-world situation. So, as I said, what I'd like to do is give you two examples of projects in my lab currently using genome editing to try to address some of these mechanistic questions. The first one will deal with a repressor of AHR function, which you've heard a little bit about already. The second one will talk about the case of evolved resistance leading to changes in the AHR signaling pathway in fish following long-term exposure. Now, the AHR repressor is it's an AHR-related protein. It represses not only the AHR, but other proteins, including HIF, hypoxia-inducible factors. It's regulated by the AHR, so it forms a negative feedback loop and serves to control AHR function. It's also is known as a tumor suppressor gene and can regulate inflammatory processes. AHRR also has clinical relevance. A common protein polymorphism in AHR repressor in humans has been linked to infertility and endometriosis. And the AHR repressor promoter is hypomethylated in newborns associated with maternal smoking. So we hypothesized that the AHR repressor may have a role in controlling AHR signaling during normal developmental processes. And so to test that hypothesis, we initially used a knockdown approach, knocking down AHR repressor A in zebrafish using a morpholino-modified antisense oligonucleotide approach. And what we found surprised us, and that was that knockdown of AHR repressor A, but not its paralog repressor B, in the absence of dioxin exposure, caused phenotypes that looked very much like the phenotypes that we see when zebrafish embryos are exposed to dioxin. That is the presence of pericardial and yolk sac edema and these craniofacial defects, including a shortened jaw. Importantly, we saw this not only with one morpholino, but with a second morpholino that was also targeted to the repressor A RNA, and that demonstrated the specificity of the effect. We also looked at gene expression in these knockdown embryos and found a dramatic decrease in expression of a number of genes involved in phototransduction in the eye, and these included a number of opsins and eye-specific phosphodiesterases. Now, when genome editing technologies came online, we immediately started to try to confirm this using initially zinc finger nuclease approach. We targeted exon 3, the repressor A gene with a ZFN, and this produced a 7 base pair deletion that led to a premature stop codon in a truncated protein that's lacking most of the functional domains of the AHR repressor. Now, the homozygous AHRRa mutants were completely viable, and surprisingly, we also showed that this was a complete loss of function allele in a transient transfection assay, unlike the wild type repressor A, which represses AHR, TCDD-inducible AHR signaling. The mutant failed to repress AHR. Now, we were surprised to find that in these AHR repressor A mutants, they were completely viable, and there were no signs of TCDD toxicity. This is showing yolk sac edema, and the repressor mutants are down here, but yet these mutants respond normally to TCDD with the induction of toxicity. When we looked at the gene expression changes, we saw the same kinds of changes in the knockouts in the phototransduction genes as we had seen in the knockdowns. So this is a situation where phenotypes induced by knockdown of a gene were not reproduced completely by the corresponding mutant. And I'm going to come back to this toward the end of the talk as one of the challenges in this field. Now, the second example involves alterations in the AHR signaling pathway as a result of evolved resistance to dioxin-like compounds in the environment. And in this case, we're testing hypotheses that were generated by population-level genomic sequencing studies. So this study involves a small estuarine fish, Fundulus heteroclitus, also known as the Atlantic killifish. And it's interesting because populations up and down the East Coast have evolved tolerance or resistance to the high levels of aromatic hydrocarbons that these fish are exposed to in the environment, including PCBs, PAHs, and dioxins. To try to identify the genes that might underlie these changes in sensitivity, we looked at 8 populations of fish, 4 tolerant sensitive pairs, and this was done in collaboration with the Fundulus Genomics Consortium. And we sequenced the genomes of 48 fish per population, and then looked for evidence of genes under selection in the tolerant populations. We found a number of outlier regions or regions under selection, and some of them are shown in red here. Several of the most highly ranked outliers were shared among all 4 tolerant populations. And these included a tandem pair of AHRs, AHR1A and 2A, an AHR target gene, CYP1A, AIP, which is a chaperone AHR interacting protein, and another pair of AHRs were under selection in 2 of the 4 populations. So the interesting result from this study was that it was not just the AHRs that were under selection, which is what we had expected to see, but other components of the AHR pathway, including especially this interesting protein AIP, which was under selection in all 4 of the populations. And so this study raised the intriguing possibility that the mechanism of resistance may not involve a single gene, but might involve epistatic interactions among several members of this pathway, and particularly between AIP and some of the AHRs. So we set out to try to better understand the role of AIP in AHR signaling in fish, and how AIP might be altered in the resistant populations, and how it might interact with AHR in an epistatic way. And so for this study, we're using both killifish and zebrafish. We're looking at all the AHRs and AIP, and we're doing not only loss of function, but also some knock-ins where we're inserting SNPs found in the killifish AIP into zebrafish so we can test their function. All right, this is the process that we go through to try to use CRISPR to target these genes in fish, either killifish or zebrafish. We inject the guide RNA and Cas9 protein into single-celled embryos. After confirming that the guide RNA is effective, we raise these to adulthood so we can screen for founders. This takes 3 months to raise zebrafish to adulthood. It takes 2 years to raise killifish to adulthood. Once we've identified founders, we then raise the F1s to adulthood, so another 2 years for the killifish. We then genotype the F1s, identify the heterozygotes, and breed them to generate homozygotes, where we can finally test them for sensitivity to PCBs. So this whole process in zebrafish can be done in about a year. For killifish, it's a 5-year project. So one of the considerations, of course, that we've talked about earlier today is the possibility of off-target effects. This becomes particularly important when you have gene duplicates or paralogs that are closely related. And so one of the things we did was to look for the possibility of off-target effects in some of the other AHR loci in these fish. In this case, I'm showing fish injected with the AHR2B guide RNA. We amplified the regions of all of the AHRs corresponding to that target site, and did surveyor endonuclease activity to look for evidence of mutations in those other sites. We did see some evidence for mismatches, but these occur in the uninjected embryos in addition to the injected embryos. And when we sequence these, we find that they correspond to known SNPs in the killifish genome. So we saw no evidence for detectable off-target effects at these related AHR loci in these fish. Now, I mentioned that we're also doing knock-in studies. The gene and protein structure of killifish AIP is very similar to the structures of the human orthologs. We've been able to identify multiple SNPs in the killifish AIP protein, and several of these occur in these functionally important TPR repeats in AIP. We're especially interested in these 2 SNPs because they occur in residues that are completely conserved in mammalian AIP as well as in zebrafish and in killifish from the sensitive populations. But in killifish from the resistant population, we see these alternative residues in these 2 sites, in one or the other of these 2 sites. And homology modeling of the killifish AIP protein predicts that these residues are important for maintaining the structural stability of this alpha helix. So we're in the process of introducing each of these SNPs individually into zebrafish so that we can test their function, because we don't want to wait 5 years to introduce them into sensitive killifish. Now, I want to finish up by just talking about some of the challenges in applying genome editing tools to developmental toxicology. Off-target effects we've talked about. These can be minimized and they can be controlled for, so I won't say anything more about them. In non-traditional model organisms, things like the long generation times and the extensive polymorphism that you see in some of these non-traditional species can be an obstacle to applying genome editing techniques. Most interesting, I think, is a discrepancy between results obtained from knockdown studies previously with more recent studies where the corresponding gene has been knocked out. And some of this might be explained by a recently introduced phenomenon of genetic compensation. So let me just say a little bit about that. This is one particular study from Nathan Lawson's lab where they compared the morpholino-induced knockdown phenotypes in zebrafish embryos for a number of genes with the corresponding knockouts, and they found a very poor correspondence in that many of the knockdown phenotypes could not be reproduced by a knockout. Now, clearly, some of these are likely to be due to off-target effects of the morpholino knockdown that weren't properly controlled for. But this doesn't explain all of the discrepancy that they saw. So more recently, Didier Stanier's group suggested and provided some evidence that there's a genetic compensation that can occur after complete knockout of a gene that's not seen after a partial loss of function or a knockdown. So there are a number of sort of compensatory mechanisms that you might imagine that could occur in a complete loss of function. Redundancy with other proteins in the genome is something that's already well known. More interesting is the possibility that lesions in the DNA caused by targeted mutation could introduce changes in chromatin structure or in non-coding RNA expression that might lead to compensatory responses, and there's a little bit of evidence for this. Even more intriguing is the idea that the mutant mRNA itself can trigger compensatory responses, and there are now several of examples of where this has occurred. For example, where exon skipping restores the reading frame after a frame shift mutation following CRISPR. Alternative splicing that can enhance the activity of an inactive paralog, so that provides partial compensation. The use of cryptic splice sites. Nonsense-mediated decay turns out to be important in some cases and required for the compensatory response to occur. And altered function of RNA binding proteins. So I think these mechanisms are not yet well understood, but I think it points to a real need to understand some of these compensatory processes that might be occurring, and not just in fish embryos, but in any kind of an organism or cell line where a gene has been completely knocked out. Now, I want to finish with highlighting what I think is one of the most exciting opportunities for applying genome editing in developmental toxicology, and that is in understanding the mechanisms underlying the developmental origins of human health and disease. And as many of you know, this refers to the idea that early life exposure to chemicals through altered fetal programming or altered epigenetic marks can lead to changes that don't become evident until much later in life as increased susceptibility to a variety of diseases, and they can even be passed on potentially transgenerationally. So I think genome editing technologies have a number of roles to play at trying to tease out these mechanisms of these two events where the exposure to the chemical and the phenotype are separated by a large amount of time. Certainly we can now do early life editing of the epigenome and look to see what effect that might have in a targeted way on downstream phenotypes later in life. Similarly, with editing non-coding RNAs early in development, the roles of DNA methyltransferases and histone modifiers can now be elucidated using genome editing. And I think perhaps most exciting is the concept of doing fate mapping or lineage tracing using genome editing techniques combined with deep sequencing. And even beyond that, actually biologically recording chemical effects occurring in individual cells. And I think we're going to hear about that tomorrow morning from Dr. Wang. But to me, this is one of the most exciting opportunities provided by genome editing in developmental toxicology. So I will end there by thanking a variety of colleagues and collaborators who've contributed to this, as well as the National Institute of Environmental Health Sciences for funding. Thank you.